This is Terpenes found in cannabis sativa are cannabinoid receptor agonists by me, Ryan Hecksel, in the Department of Pharmacology in the lab of Dr. John Stryker. We're going to keep this plant theme going we got today. I don't directly work with cannabis sativa. I only work with terpenes. We're going to talk about what terpenes are, where they're found in cannabis, and what is an agonist. First, what is a terpene? Well, they're a diverse class of organic chemicals, primarily hydrocarbons. Occasionally, we'll get a hydroxyl group in there. They have diverse smells. Beta-pinene is aptly named. It smells like pine trees. It's also found in pine trees. Beta-carotene is found in hops. Geraniol is found in rose oil. Terpenes are the basis of essential oils, and typically they're used in perfumes for their strong smell. Where are terpenes found in cannabis? Are they found in the leaves? No, they're not. They're found here in the flower. And if we zoom in closer on the flower, we'll see these trichome structures. And these are glandular structures in plants that secrete terpenes and cannabinoids like CBD and THC. Now, these aren't the only terpenes found in cannabis. It varies greatly by strain, but these are the ones we wanted to take a look at for our research. Let's go into some basics of pharmacology research. So if we wanted to test if a compound is a drug that acts at a receptor, we're going to administer it, and we're going to see a signal transduction pathway, which ultimately causes a cellular response. So if we wanted to take a look at if a compound was a drug, we would look for certain proteins and signaling molecules in this signal transduction pathway. That's exactly what we did. We took a cell line overexpressing the cannabinoid type 1 receptor, which is the same receptor that THC binds to. And we found that beta-pinene, alpha-humulene, linalool, and beta-carotene cause significant levels of ERK phosphorylation. You can see one of my Western blots down here, which could be abolished or reduced. ERK phosphorylation could be reduced with administration of rimonabant. Now, this would block the CB1 receptor and not allow interaction with the terpene. So this suggests that the CB1 receptor is essential to see this ERK phosphorylation. We also saw ERK phosphorylation in cannabinoid type 2 receptor, which is primarily what CBD interacts with. We also took a wild type cell line overexpressing no one particular receptor, and we saw that alpha-humulene, beta-pinene, and beta-carotene caused ERK phosphorylation, which could not be reduced by rimonabant, suggesting that there's a different target, different receptor that these terpenes are able to interact with. We also saw cyclic AMP inhibition, which further suggests a cannabinoid-based mechanism. So we took these compounds and we did some in vivo work. Here, we're only going to look at alpha-humulene administration in mice. The greater the thermal latency here, the greater the analgesic efficacy of a compound, or the greater the pain relief of a compound. So over about a course of an hour, alpha-humulene caused significant levels of analgesia. It also caused a body temperature decrease. It also caused catalepsy and hypolocomotion. These four symptoms or behaviors in mice are known as the tetrad test, and they suggest a cannabinoid-based mechanism as well. Where do we go from here? Where do we go from this basic science? So we've seen that across the United States, many states have legalized cannabis for medical use. However, cannabis as a medicine is an inexact science because we don't really know everything about it. We don't know everything in it. So we've identified that terpenes are able to cause analgesia. Hopefully, we can identify a terpene like alpha-humulene, who's very good at causing analgesia compared to the other terpenes. And hopefully, we can identify more about the mechanism and how they're working to better produce medical marijuana strains as alternatives to opioid therapy. And I'd like to thank my PI, John Stryker, grad student I used to work with, Justin, and my postdoc, Attila, for all their hard work.